Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to cover chapter 6 in our MCAT Biochemistry series, and this chapter is titled DNA and Biotechnology. In this chapter, we're going to cover the following objectives. First, we're going to review the structure of DNA, starting with the building blocks called nucleosides and nucleotides. Then we're going to move on to how these link together through the sugar-phosphate backbone. We're also going to discuss purines and pyrimidines, and then we're going to walk through the Watson-Crick model of the DNA double helix. Finally, we're going to explain what it means for DNA to denature and reanneal. Next, we're going to explore how DNA is packaged inside cells by diving into eukaryotic chromosome organization. We're going to talk about histones, the difference between heterochromatin and euchromatin, and what roles telomeres and centromeres play. Third, we're going to move on to DNA replication. So here we're going to cover how strands separate, where replication begins, and how daughter strands are synthesized. We're also going to discuss what makes the ends of DNA strands tricky to replicate and how cells deal with that. After that, we're going to look at DNA repair mechanisms. This is going to include the role of oncogenes and tumor suppressor genes in cancer, as well as repair processes like proofreading, mismatch repair, nucleotide excision repair, and base excision repair. Finally, we're going to wrap up with recombinant DNA and biotechnology, where we're going to look at cloning, restriction enzymes, DNA libraries, and cDNA. We're also going to cover techniques like PCR and southern blotting, as well as DNA sequencing. Then we're going to go through applications of DNA technology and make a final note on safety and ethics. With this introduction in place, let's go ahead and get started with objective 1. First, both DNA and RNA fall under the category of nucleic acids. These are long molecules that carry genetic information. And while they're made of similar types of building blocks, they do play very different roles in the cell. In this chapter, we're going to focus specifically on DNA. In the next chapter, we're going to focus on RNA. So, with DNA as our goal for this chapter, let's begin. DNA is a polymer. That means it's made of many repeating units strung together. These repeating units are called monodeoxyribonucleotides. That word might sound intimidating at first, but it just means that DNA is made of individual building blocks, and each of these contains 3 key parts: a sugar, a nitrogenous base, and 1 or more phosphate groups. Now, the first part of each building block is a 5-carbon sugar. This is also called a pentose sugar. This sugar is important because it gives the entire molecule directionality, and it also helps determine whether we're talking about DNA or RNA. Now, if we go ahead and we attach a nitrogenous base to that sugar, specifically at carbon 1, we've made what's called a nucleoside. So a nucleoside is just a sugar plus a nitrogenous base. That's it. Now, if we go ahead and we add 1 or more phosphate groups to that structure, bonded to carbon number 5 on that sugar, now we've created a nucleotide. So here's a quick summary. A nucleoside is a sugar plus a base. A nucleotide is a nucleoside with phosphate groups attached to it. Now, these nucleotides are the true building blocks of DNA. And when they're strung together in a long chain, they form the DNA polymer. And these phosphate groups, they're not just structural. They also carry negative charges, and those negative charges are gonna repel each other. That repulsion actually stores potential energy, which is why nucleotides like ATP are used as energy carriers in the cell. Now, before we jump into the specific names and details of the nitrogenous bases, I want to step back and I want to address something important. How do we tell DNA and RNA apart? Even though we're not focusing on RNA in this chapter, it's important to make that distinction. And the answer lies in the sugar. Both DNA and RNA contain this 5-carbon sugar. But there is one small difference between them, and it has big consequences. In RNA, the sugar is a ribose. Ribose has a hydroxyl group, an OH group, attached to carbon number 2. In DNA, the sugar is deoxyribose, and here that same carbon just has a hydrogen atom instead of the hydroxyl group. That's what deoxy means, lacking oxygen. So this missing hydroxyl group makes DNA more stable than RNA. RNA's extra hydroxyl group makes it more reactive and more easily broken down. That's why DNA is used to store long-term genetic information, while RNA is used for more temporary tasks like protein synthesis. And this distinction is something you should absolutely be able to recognize visually on the MCAT. So if you're shown a sugar structure and you're asked whether it's part of DNA or RNA, look for the hydroxyl group on carbon 2. If it's there, you're looking at RNA, and if it's not there, you are looking at DNA. Now, what you're seeing on this part of the page is a summary of the nomenclature of important bases, nucleosides, and nucleotides. Here in this first column, we see a list of the 5 key nitrogenous bases: adenine, guanine, cytosine, uracil, and thymine. And it shows how their names change when they're incorporated into nucleosides or nucleotides. This is great to review and get familiar with for test day, but this chart will actually make a lot more sense once we understand how DNA is built and how these nitrogenous bases fit into the larger structure. So let's come back to this later. Let's instead discuss the backbone of DNA. The DNA molecule, it's made up of a long chain of nucleotides, but it's not the bases that form the chain. It's the sugar and phosphate groups that link together to form what we call the sugar-phosphate backbone. This backbone is very important because it gives the DNA strand directionality, which means we always read and build it in a specific direction, from the 5' end to the 3' end. Now, where do those numbers come from? They refer to the carbon positions on the sugar. The 5' carbon is where the phosphate group attaches, and the 3' carbon is where the next nucleotide connects. The bond that forms between them is called a phosphodiester bond. This bond links the 3' hydroxyl group of one sugar to the 5' phosphate group of the next nucleotide. And this linkage repeats again and again, and it forms a strong, stable backbone with all of the nitrogenous bases sticking out to the side, like rungs on a ladder. Now, because each phosphate group carries a negative charge, the entire DNA molecule has an overall negative charge. And this becomes important later when we talk about how DNA interacts with positively charged proteins like histines, or when we use gel electrophoresis to separate DNA fragments. So just keep in mind for now that DNA carries an overall negative charge, and that has a lot of important consequences. Now, DNA in cells isn't just a single strand. Okay, it's not just a single strand. It's almost always found as a double-stranded molecule. These two strands wrap around each other in a spiral, forming the famous double helix. We're going to come back to how we reach this conclusion in a little bit. But building on that, one strand is going to read from the 5' to the 3' direction. The other strand that's attached to form this double helix, it runs from the 3' to the 5'. And this is anti-parallel. So the other strand runs anti-parallel, meaning it goes 3' to 5'. And this anti-parallel orientation is really important for base pairing, replication, and transcription, which we're going to get into soon. Now, let's go ahead and talk about the nitrogenous bases. There are two main families of nitrogenous bases that are found in nucleotides: purines and pyrimidines. Purines are made of two rings that are fused together, a 6-membered ring and a 5-membered ring. The two purines you need to know are adenine and guanine, and they are both found in DNA. On the other hand, we have pyrimidines. These are smaller. They contain just one 6-membered ring. The 3 pyrimidines that you need to know are cytosine, thymine, and uracil. Now, one of these, cytosine, is found in both DNA and RNA. Thymine is only found in DNA, and uracil is only found in RNA. Now, from a chemistry standpoint, both purines and pyrimidines are considered aromatic heterocycles. A heterocycle is simply a ring structure that includes atoms other than carbon. So in this case, nitrogen. And aromatic means that the ring is stabilized by a special kind of electron delocalization that follows a specific rule called Huckel's rule. Now, we discussed this further in the MCAT organic chemistry playlist, but we're still going to review it here. To be aromatic, a molecule has to meet 4 conditions. The first condition is that it has to be cyclic. In other words, form a closed ring. The second condition is that it has to be planar or flat so that all of the atoms lie in the same plane. Third, it has to be conjugated, meaning it has alternating single and double bonds, or it has lone pairs that allow for electron delocalization, conjugated. Fourth, and the final rule, is that it has to have a specific number of pi electrons following the formula 4n+2. That's Huckel's rule. Now, all of the nitrogenous bases in DNA and RNA, guess what? They meet these criteria, and their aromaticity contributes to the stability of the DNA molecule through something called base stacking. This helps hold the double helix together in addition to hydrogen bonding. Now that we've seen the base structures and you know where they attach on the sugar and how the backbone is formed, we can go back and look at the chart of nucleoside and nucleotide names. So I'm going to scroll all the way back up, and now we're going to work through this table very briefly because we have all the background information we need to begin to understand it. You'll notice here that naming is based on 2 things: the identity of the nitrogenous base and the number of phosphate groups that are attached. We're going to focus on adenine to work through the table, and then the same principles extend to all of the nitrogenous bases here. So for adenine, let's start with nucleosides. When adenine is attached to ribose, we call it adenosine. When it's attached to deoxyribose, it becomes deoxyadenosine. Now, once we add phosphate groups to the nucleoside, we form nucleotides. And the names follow a clear pattern based on the number of phosphates. So for the ribose version, if there is 1 phosphate, we have adenosine monophosphate, AMP. If we have 2 phosphates, we have adenosine diphosphate, or ADP. And if there's 3 phosphates, then it's adenosine triphosphate, or ATP. For the deoxyribose version, if we have 1 phosphate, we have deoxyadenosine monophosphate. For 2 phosphates, we have deoxyadenosine diphosphate. And for 3 phosphates, it's deoxyadenosine triphosphate. And once again, the same idea extends to the other nitrogenous bases in this table. Now that you know how to read and understand this table, I'm going to scroll back down here and just provide a summary of everything we've discussed up until this point. So this image summarizes DNA structure by showing the nucleotide components. We have a phosphate group, a deoxyribose sugar, and we know that's the case because at carbon 2, there's only hydrogens. And then there is a nitrogenous base. The phosphate is attached at carbon 5. The nitrogenous base is attached at carbon 1. Now, the nitrogenous bases, they include purines like adenine and guanine. And you'll notice that purines, they have 2 ring structures. And then the other category of nitrogenous bases we discussed are pyrimidines. And for DNA, that includes cytosine and thymine. These are 1 ring structures. Nucleotides, they link through phosphodiester bonds between the 3' hydroxyl group and the 5' phosphate groups, and they form the sugar phosphate backbone. And the sequence of bases, they encode genetic information. So now everything fits together, the sugar, the base, the phosphates, and how they come together to form the full structure of DNA. With all of this new information in mind, we're ready to ask the million-dollar question. How did we figure out what DNA looks like? In the 1950s, James Watson and Francis Crick used X-ray diffraction data, originally produced by Rosalind Franklin, to develop a physical model of DNA. And that model became the double helix we know today. Now, before we discuss the details of this model, can I share some tea about Mr. James Watson that I think we should all know? And you might want to sit down for this one. So, he made many racist and sexist remarks over the year. I know, shocker, a white man saying something wrong? Say what? Now, in a 2007 interview, and don't quote me on the date, he said that people of African descent were inherently less intelligent than others. And that is not only scientifically absurd and wrong, but deeply, deeply racist. Jail immediately. And alas, it gets worse. This man literally doubled down on those views in a 2019 documentary, and that actually resulted in him getting his honorary titles revoked. At some point in between, this man had to auction his Nobel Prize medal because he was running low on cash. And the medal actually sold for like over $4 million. And the person who bought it actually later returned it to him. So the buyer must also be racist. Very lame. To top it all off, we know how much Rosalind Franklin's role was historically minimized, in part because of gender bias. The X-ray crystallography data that Watson and Crick literally used to build their model, it came from her lab, and they saw it without her explicit permission. And I think we call that theft. Anyways, now that we know how terrible these men are, let's go over the key features of the DNA model step by step. First, DNA is a double helix. That means the 2 strands of DNA, they spiral around a shared axis, kind of like a twisted ladder. And this helical shape is really important because it allows for compact storage, and it very much contributes to structural stability. Second, the 2 strands, they run anti-parallel. Like the 2 strands are anti-parallel, like we mentioned earlier. What does that mean? One strand is going to run from the 5' to the 3' direction, and the other strand runs 3' to 5'. Third, the sugar phosphate backbone of each strand, it's going to be on the outside of the helix. It forms the sides of the ladder and it provides that structural support. Then on the inside, right, this leads us to the 4th point. Nitrogenous bases, they stick inward like the rungs of a ladder. And these are what actually interact across the two strands. And that brings us to base pairing. Watson and Crick, they proposed specific complementary base pairings, which were later confirmed by Chargaff's rules. And here's how it works. Adenine always pairs with thymine through two hydrogen bonds. We can see that right here. Here's T, here's A. They pair through two hydrogen bonds. Guanine, it always pairs with cytosine through three hydrogen bonds. And we can see that right here. G pairs with C, and we have three hydrogen bonds. These specific pairings are what allow one DNA strand to actually determine the sequence of its partner strand because we know what pairs with what. And here's what Chargaff discovered. In any double-stranded DNA molecule, the amount of A always equals the amount of T, and then the amount of G always equals the amount of C. So that means the total number of purines, A and G, that's going to be equal to the total number of pyrimidines, C and T. But hydrogen bonds, they aren't the only thing that's stabilizing the helix. The structure is also stabilized by something called base stacking. This refers to the van der Waals interactions between the flat aromatic bases as they stack on top of each other inside the helix. So these interactions are also going to add some extra stability to the overall structure. So just to wrap up and give you the takeaway points, the DNA double helix is made of two anti-parallel strands. The sugar phosphate backbone is on the outside. The nitrogenous bases are on the inside, and they're held together by hydrogen bonds, and they're stabilized by base stacking. And the sequence of one strand determines the sequence of the other. And that's going to be the basis for DNA replication and transcription. Now, the most common form of DNA that's found in cells is called bDNA. bDNA, this is the familiar right-handed double helix that we have been talking about. In bDNA, the helix makes a complete turn every 3.4 nanometers, and there are about 10 base pairs per turn. Now, the strands, they wrap around each other in a smooth spiral, like you see here, and the molecule has major and minor grooves. These are the spaces between the backbone where proteins and other molecules can interact with the bases inside. But not all DNA adopts the B form. There's also a less common form called zDNA, and this is a left-handed helix. It's more extended and zigzag-shaped with 12 base pairs per turn and a full twist every 4.6 nanometers. Now, zDNA, it can appear under certain physiological conditions, especially when there's a high GC content or there's unusual supercoiling. But for most of our discussions, we're going to stick with bDNA as the standard. Now let's talk about something really important for both biology and biotechnology: DNA strand separation. During processes like replication and transcription, the two strands of DNA, they have to be separated to allow enzymes to access the genetic code. And this process of separating the strands is something called denaturation. Denaturation happens when the hydrogen bonds between the base pairs break, usually because of things like heat or changes in pH or the use of a chemical agent. This results in single-stranded DNA, which is necessary for enzymes like DNA polymerase or RNA polymerase to work. Once the denaturing conditions are removed, though, like when you let the sample cool down, for example, those single strands, they can actually come back together through a process called reannealing. Reannealing is simply the reformation of the hydrogen bonds between complementary bases, and that's going to restore the double-stranded structure. Now, these concepts, denaturation and reannealing, they are the foundation of a very important lab technique called polymerase chain reaction, or PCR. In PCR, we take advantage of this cycle of separating and then repairing DNA strands. And first, we heat the sample to denature the DNA, and then we cool it so that short DNA primers can bind to their complementary sequences. Finally, a special DNA polymerase is going to extend the primers to synthesize new DNA strands. And this cycle repeats over and over to amplify a specific DNA sequence. Now, this is a very brief overview of PCR. We're actually going to go into a lot more detail about PCR later in this chapter. But for now, I want you to remember denaturation and reannealing, they're not just biological processes, they're also tools that we're going to use in the lab to manipulate DNA. With that, we've completed objective 1. We can now move into objective 2. Here we're going to talk about how DNA is actually packaged and organized inside eukaryotic cells. Eukaryotic DNA doesn't just float freely in the nucleus. It's actually carefully wrapped, folded, and compacted into structures we call chromosomes. To make sense of this, we have to start with the basics. DNA contains regions called genes, and these encode proteins and other functional molecules. In humans, all of this genetic information is divided up among 46 chromosomes, which are found in the nucleus of each cell. But how do you fit over 2 meters of DNA into a microscopic nucleus? That's where chromatin structure comes in. The first level of DNA organization involves histone proteins. These are small, positively charged proteins that help DNA wrap tightly without tangling. DNA winds around a core that's made of 8 histone subunits, 2 copies of H2A, 2 copies of H2B, H3, and H4, and they form a structure called a nucleosome. Now you can think of nucleosomes like beads on a string, with each bead being a segment of DNA that's wrapped around histones, and the string being the linker DNA between them. And this arrangement really helps condense DNA. Now, a separate histone called H1, it's going to help stabilize this structure, and it's going to promote further packing into more compact chromatin fibers. As chromatin gets more and more tightly coiled and structured, it eventually becomes visible under a microscope as a chromosome, especially during cell division. Now, not all chromatin is equally accessible. This brings us to the concepts of heterochromatin and euchromatin. Heterochromatin is more tightly packed form of DNA. It's dense, it's transcriptionally silent, and it's going to appear dark under a light microscope. So that means the genes in heterochromatin are not actively being used. They're shut down either temporarily or permanently. Euchromatin, on the other hand, it's loosely packed, it's more open, and it's transcriptionally active. So it's going to appear lighter under a microscope, and this is where you'll find genes that are currently being expressed. In other words, genes that are being transcribed into RNA. So here's an easy way to remember this. Heterochromatin is dark, dense, and silent. Euchromatin is light, loose, and expressed. From here, we want to move into discussing telomeres and centromeres. Telomeres are located at the very ends of chromosomes, and their job is really to protect the ends of DNA and prevent it from unraveling. And they do this by having a high GC content, guanine and cytosine, and they form 3 hydrogen bonds. And that's going to be stronger and more stable than AT pairs. That's why telomeres have a high GC content. And this stability really helps telomeres kind of act like caps on the chromosome ends. However, they do face a problem. Every time a cell divides, they get a little bit shorter. And that's because DNA polymerase, which we're going to see in the next objective, they can't fully replicate the very end of the lagging strand. So over time, this shortening of the telomeres, it contributes to cellular aging and eventually cell death. But cells can counteract this using an enzyme called telomerase, and it adds these repeating sequences back to the telomeres. This enzyme is active in stem cells, germ cells, and also, unfortunately, often in cancer cells. As for centromeres, these are regions that are found near the middle of chromosomes, and their main role is to hold sister chromatids together after DNA has been replicated. Now, during anaphase of mitosis, the centromere is where spindle fibers attach, so the chromatids can be pulled apart into separate cells. If you want more information about this, it's going to be in chapter 2 in the MCAT biology playlist in case you need to touch up on mitosis and the steps of mitosis. Now, like telomeres, centromeres also contain high GC content, which gives them structural strength and it helps them remain tightly bound. So together, telomeres and centromeres are very much essential for maintaining the integrity of chromosomes, especially during cell division. Now, objective 2 was nice and simple. Onwards to objective 3. Here we're going to discuss DNA replication. Now, before we walk through the entire process step by step, we're going to first introduce the key players that are involved in replication. And once we've laid out the parts, we're going to see how they come together to carry out DNA replication. Now, in this objective, there's going to be a lot of repetition, but you're going to sit down and listen, and you're going to like it. So, starting from the top, the first step in replication is identifying the origin of replication. This is going to be the specific site on the DNA where replication begins. So in prokaryotic cells like bacteria, there's only going to be one origin of replication per chromosome. But in eukaryotic cells, which have much larger genomes, there's going to be multiple origins of replication per chromosome. And that's going to allow replication to occur more quickly across long stretches of DNA. Once replication begins, the DNA double helix is going to have to be unwound. And this job is carried out by an enzyme called helicase. Helicase breaks the hydrogen bonds between complementary bases, and it's going to separate the two strands of DNA and create what we call a replication fork on either side of the origin. Now, as helicase unwinds the DNA, it creates exposed single strands, but these strands are going to be unstable and they could snap back together. So to keep them open, single-stranded DNA binding proteins attach to each strand and hold them apart. Next, the enzyme primase comes in to lay down short segments of RNA primers. These primers are necessary because DNA polymerase, which is the enzyme that builds new DNA, it can't start from scratch. It can only add nucleotides to an existing strand. So primase gives DNA polymerase a starting point. Now that the site is open and primers are in place, DNA synthesis can begin. In prokaryotes, this job is carried out primarily by DNA polymerase III. In eukaryotes, there's multiple polymerases that are involved. The most important are DNA polymerase alpha, delta, and epsilon. Now, as the new DNA is being synthesized, the RNA primers that primase lays down, they need to be removed. So in prokaryotes, DNA polymerase I is going to do that job. It's going to remove the RNA primers using its 5' to 3' exonuclease activity. In eukaryotes, this task is performed by an enzyme called RNase H, which also works in the 5' to 3' direction. Once the RNA primers are removed, they can actually now be replaced with DNA nucleotides. So in prokaryotes, this is handled again by DNA polymerase I, and in eukaryotes, it's going to be handled by DNA polymerase delta. Next, those small segments of newly made DNA, especially on the lagging strand, they're going to need to be joined together. So these fragments are called Okazaki fragments, and the enzyme that seals them is going to be called DNA ligase. DNA ligase forms phosphodiester bonds between adjacent fragments to complete the strand. Now, one more complication occurs as helicase continues to unwind DNA, and that complication is that it introduces positive supercoils ahead of that replication fork. And these coils make the DNA tighter and a little bit harder to unwind. So to relieve this tension, cells use DNA topoisomerase. In prokaryotes, this is often referred to as DNA gyrase. But topoisomerases, they cut the DNA, they allow it to unwind, and then they reseal it, essentially relaxing the strain. Finally, there's one step that applies only to eukaryotes, not prokaryotes, and that is the synthesis of telomeres. So like we mentioned earlier, DNA polymerase, it can't fully replicate the ends of linear DNA. Without a solution, that would lead to gradual loss of genetic material over time. So to solve this problem, eukaryotic cells have help from the enzyme telomerase. This is an enzyme that extends the ends of chromosomes using its own RNA template. In prokaryotes, this step is not necessary because the chromosomes are circular. They don't have ends to lose. Now that we've gone over all of the enzymes and the proteins that are involved, let's walk through the process in order and see how all of these pieces really come together and visualize it. Again, this might seem repetitive, but I think for this concept, it's going to be really important. The entire DNA replication process is carried out by a multi-protein structure known as the replisome or replication complex. And that includes all of these enzymes that we talked about that work together to unwind the DNA, to copy the strands, and to finalize the new daughter molecules. Now, everything it begins at a specific site on the DNA called the origin of replication. In prokaryotic cells, that's typically just one origin per chromosome. But in eukaryotic cells, there's actually multiple origins per chromosome to really allow for faster replication of this larger genome. Now, once the origin is identified, helicase attaches to the DNA and it begins to unwind the double helix. You can see that illustrated in the center of the diagram by this blue wedge-shaped protein. Now, as helicase moves forward, it breaks the hydrogen bonds between base pairs and it exposes two single-stranded DNA templates. Now, this unwinding action, it really introduces tension ahead of the fork, and that is going to be relieved by topoisomerase. That's shown in this large green ring on the right. Now, topoisomerase, it makes temporary cuts in the DNA to allow it to relax and to prevent supercoiling before resealing it. As the DNA is being unwinded, single-stranded DNA binding proteins, these are the small purple balls in the image, they're going to quickly coat each strand. These proteins are going to keep the strands from rejoining, from reannealing, and it protects them from degradation. Now that the DNA is open, replication can begin. First, the cell needs to provide a starting point. That's where primase comes in. It's shown here in teal. Primase synthesizes short RNA primers. So these are short sequences that provide a free 3' hydroxyl group for DNA polymerase to build upon. And once these primers are laid down, DNA synthesis begins. Now, at each replication fork, we have two template strands. One that's oriented from 3' to 5', and the other one from 5' to 3'. Because DNA polymerase can only synthesize new DNA in the 5' to the 3' direction, the two new strands are made in different ways. So we're going to start with the leading strand. Because The template strand for the leading strand runs 3' to 5' towards the replication fork. All right, DNA polymerase can actually follow right behind helicase and synthesize the new strand continuously. This is carried out by DNA polymerase. Meanwhile, the other template strand, the lagging strand, it's oriented in the 5' to the 3' direction, which is actually opposite to how DNA polymerase operates. So for the lagging strand, we can't synthesize the new strand from this in a continuous manner. So instead, it's built in short fragments known as Okazaki fragments. And here's how this works. Primase periodically lays down new RNA primers closer and closer to the replication fork. Then DNA polymerase alpha adds a short stretch of DNA to each primer, and DNA polymerase delta takes over to extend the fragment. Each fragment is synthesized in the 5' to the 3' direction, but overall the fragments are made away from the replication fork. Eventually, those RNA primers on the lagging strand, they have to be removed. So after they are removed, then DNA polymerase delta goes ahead and fills in the gaps with DNA nucleotides. The last step is obviously to seal things between the fragments. This is the job of DNA ligase. It's seen here as this orange block. It forms phosphodiester bonds between adjacent fragments to create a strand, to create a continuous new strand. So all of this activity, unwinding, priming, synthesis, everything, it occurs at both replication forks on both the leading and the lagging strands. And it repeats across multiple replication bubbles in eukaryotic genomes. And this very tightly coordinated process, it makes sure that both strands of DNA are copied quickly and accurately and produces two identical daughter DNA molecules, each consisting of one old strand and one newly synthesized strand. And this is what we call semi-conservative replication. So I just want to repeat this because I want you to remember this. When we are looking at the leading strand, all right, the newly synthesized strand from the leading strand is created continuously, okay? Because the template strand for the leading strand runs 3' to 5' towards the replication fork, DNA polymerase can follow right behind and synthesize that new strand continuously. Meanwhile, the other template strand, the lagging strand, that can't happen. So it is not continuous. And that is the process that we work through in detail. With that, we have completed objective 3. We are now going to move into objective 4, which is all about DNA repair. First, let's talk about why DNA repair is even necessary. The structure of DNA, it can be damaged in many different ways. Some of this damage happens during replication, while other damage occurs randomly due to environmental exposures like UV radiation, chemicals, or even reactive oxygen species that are produced during normal metabolism. When DNA gets damaged and it isn't repaired, that can lead to mutations. And some of those mutations can result in uncontrolled cell growth and ultimately cancer. On that note, let's break this down further by talking about the two key types of genes that are involved in cancer: oncogenes and tumor suppressor genes. Oncogenes are mutated versions of normal genes called proto-oncogenes. Proto-oncogenes normally help regulate cell growth and division, but when they're mutated, they can become overactive and they can drive unchecked cell division. In other words, a single mutation can turn a proto-oncogene into an oncogene, and that can tip a normal cell into cancerous behavior. On the other hand, tumor suppressor genes like P53, they can act as safeguards. They work by halting the cell cycle or by triggering DNA repair when damage is detected. Because of this, tumor suppressor genes, they're called anti-oncogenes. But if these genes are mutated and they lose function, then the cell loses that protective mechanism. And you can see that balance in this diagram. On the left, we have normal proto-oncogenes and tumor suppressor genes. They keep the cell cycle under control and they promote healthy division. But on the right, mutations in either type of gene through the loss of function in tumor suppressors or gain of function in proto-oncogenes, they can disrupt that balance and they can lead to cancer. With that in mind, let's look at how cells actually repair DNA. DNA repair falls into two main categories: replication error repair, which fixes mistakes that are made during or just after DNA replication, and damaged base repair, which corrects chemical or environmental damage that can occur at any point in the cell cycle, not just during replication. Let's start with the replication errors. There are two main mechanisms here: proofreading and mismatch repair. Proofreading happens as DNA polymerase is synthesizing the new strand. If it detects a mismatched or unstable base pair, it can pause, it can remove that incorrect nucleotide, and then it can replace it with the correct one. And this helps keep error rates low because it fixes the error right there. Then there's mismatch repair. On the other hand, it occurs after replication is complete, specifically during the G2 phase of the cell cycle. This system scans for base pairing errors that escaped proofreading, and it uses methylation to distinguish the newly synthesized strand from the template strand, and then removes the error and replaces it. The key players here include MSH2 and MLH1, which help recognize the mismatch. And then it includes proteins mu-S and mu-L, which coordinate the repair. Now, for the second category, repair of damaged bases, these pathways, they correct DNA damage from environmental or chemical sources, and they don't depend on the replication process. There are two main types of repair systems here. We have nucleotide excision repair, and we have base excision repair. Let's start with nucleotide excision repair. This is used to fix bulky lesions like thymine dimers that are caused by UV radiation. Now, in excision endonuclease, it cuts on both sides of the damaged section, and then DNA polymerase fills in the gap and DNA ligase seals it. For base excision repair, this deals with smaller, non-bulky damage, and in this pathway, we have a glycosylase that removes the damaged base, and it leaves behind an empty sugar phosphate backbone called an AP site. An AP endonuclease then cuts the backbone at that site, and DNA polymerase and ligase come in afterwards to fill and seal the gap. With that, we'll move into our last and final objective for this chapter: recombinant DNA and biotechnology. Recombinant DNA technology gives us the ability to take a DNA fragment from any source, whether human, bacterial, or viral, and amplify it, study it in the lab. There are two main approaches that we're going to discuss here: gene cloning and polymerase chain reaction, or PCR. Let's start with gene cloning. In this process, we use special enzymes called restriction enzymes, also known as restriction endonucleases, to cut DNA at specific recognition sites. These sequences are often palindromic, meaning they read the same forward and backward. Once we've cut the DNA, we can insert the desired fragment into a vector, typically a plasmid. This forms recombinant DNA, a hybrid molecule that combines DNA from two different sources. We then introduce this recombinant plasmid into bacteria, and the bacteria, they replicate this plasmid as they divide, which produces a lot of copies of the DNA. In some cases, they can even express the gene, which gives us a reliable method for producing proteins like insulin in large quantities. We can walk through this visually. It's what you see right here. We start with a plasmid and the gene of interest, say the insulin gene. Both are digested using the same restriction enzyme, and it creates these sticky ends that make it easier to pair and insert the gene into the plasmid. DNA ligase then seals the backbone and it forms a stable recombinant molecule. And once inside the bacteria, this plasmid can be replicated or used to express the gene and produce protein. To further analyze genes, we often turn to DNA libraries, which are curated collections of cloned DNA sequences that are designed for research and screening. There are two main types of DNA libraries. We have genomic libraries and we have cDNA libraries. A genomic library contains the entire DNA sequence of an organism. It includes both exons, which are coding regions, and introns, which are non-coding regions. These are comprehensive, but genomic libraries, they aren't ideal for studying gene expression because they also include DNA segments that aren't transcribed or translated. On the other hand, a cDNA library is created by reverse transcribing mRNA into DNA. So these libraries only contain the genes that were actively expressed in the original cell or tissue. No introns, just exons. That's why they're often called expression libraries. They focus on the functional genetic output. Now, another tool that's often used by researchers is called hybridization. Hybridization is the pairing of complementary nucleic acid strands, and it can occur between DNA and DNA or DNA and RNA strands. And it's essential in many molecular biology techniques. Now, with that being said, I want to discuss polymerase chain reaction, gel electrophoresis, southern blotting, and DNA sequencing. Let's start with polymerase chain reaction, or PCR. PCR is this powerful automated method that can generate millions of copies of a specific DNA sequence from just a tiny starting sample. It's extremely sensitive, it's extremely fast, and it relies on primers that hybridize to the target sequence to pretty much kickstart the amplification process. So how does PCR actually work? You're going to begin with your DNA sample, along with short primers, free nucleotides, a heat-stable enzyme called Taq polymerase, and a buffer solution. These are going to be combined in a PCR tube and placed into a thermal cycler that runs through a series of temperature changes. Each PCR cycle includes 3 steps. First, denaturation. The mixture is heated to about 95 degrees Celsius to separate the 2 strands of DNA. Then we have annealing. The temperature is lowered to around 55 degrees Celsius so that primers can bind or anneal to their complementary sequences on the DNA. Finally, we have extension. At 72 degrees Celsius, Taq polymerase synthesizes new DNA strands by adding nucleotides to the primers. And these steps, they're repeated over many cycles, which allows the DNA to double each time, and that leads to exponential amplification. But after amplification, we often want to analyze or compare DNA samples, and that's where gel electrophoresis comes in. In gel electrophoresis, DNA samples are going to be loaded into an agarose gel and then subjected to an electric field. We know DNA is negatively charged, so it migrates towards the positive electrode. Smaller fragments are going to move faster, and that's going to allow us to separate DNA by size. Now, this is discussed further in our MCAT biochemistry playlist. I believe we went over gel electrophoresis in chapter 3. So you can revisit that if you want more details. But gel electrophoresis is also often used while performing southern blot. And that's what we're going to discuss next. This process often involves transferring the DNA from the gel to a membrane and then adding a labeled probe, which is a short single-stranded DNA sequence that is complementary to the target. The probe hybridizes to the matching DNA sequence, and detection methods such as X-ray films are used to visualize the result. Now, southern blotting really allows us to detect the presence of a specific DNA fragment. And when it's properly calibrated, it can also provide semi-quantitative information about how much of the sequence is present. Now that we've covered amplification and detection, let's talk about DNA sequencing. This is the process of determining the exact order of nucleotides in a DNA strand. Now, modern sequencing methods, they use dideoxyribonucleotides, or DDNTPs, which are special nucleotides that lack a 3' hydroxyl group. Now, without this group, DNA polymerase can't continue adding nucleotides. So the DNA chain stops growing when a DDNTP is incorporated. By adding a small amount of these chain-terminating nucleotides to a normal DNA replication reaction, we can actually generate a collection of fragments of different lengths, each ending with a DDNTP that is often fluorescently labeled. Now, when we separate these fragments using something like gel electrophoresis, the pattern of fragment lengths are going to correspond to the DNA sequence, and that is going to allow us to read the genetic code base by base. So with these tools, cloning, PCR, electrophoresis, blotting, and sequencing, we have everything we need to not only amplify DNA, but also analyze, detect, and interpret it at the molecular level. Now that we've covered the core techniques in recombinant DNA, let's finish this chapter by discussing some powerful applications of this technology. First, we have gene therapy. This is a method that's used to treat or potentially cure genetic disorders by introducing a functional copy of a gene into a patient's cell. Now, typically, this is done using a viral vector. This is a virus that's been genetically modified so it can no longer reproduce or cause disease. The functional gene is inserted into the viral genome, and the virus is then introduced to patient cells, either directly or after the cells are extracted and modified in the lab. Once inside the cells, the introduced gene can then begin producing the missing or defective protein, and that can potentially correct the underlying problem. Now, this approach has been explored for disorders like severe combined immunodeficiency, certain inherited blindness conditions, and even some forms of cancer. Another major application is in transgenic animals, particularly transgenic mice. These are genetically modified mice that carry foreign genes, often inserted at the embryonic stage. Scientists use these mice to study gene function and model human diseases. Now, the inserted gene is called a transgene, and it might be one that we want to study for overexpression or mutation effects or regulation. Closely related to this is knockout mice. In these animals, a specific gene is intentionally deleted or even inactivated, and that allows us to study what happens when that gene is missing. And this is especially helpful for modeling loss of function mutations, which are common in genetic diseases. And this diagram here kind of illustrates the different approaches. In one approach, we can inject a gene into a fertilized egg or early embryos. In another, we can use embryonic stem cells that have been genetically modified in vitro, and then we inject those into developing embryos. In either case, the resulting offspring are screened to see whether the gene has been successfully integrated into their genome. Finally, as with any genetic technology, it's really important to consider safety and ethics and consent. Gene therapy, for instance, it has to be carefully controlled to avoid unintended effects like immune reactions or insertional mutagenesis. With transgenic animals, ethical considerations include the welfare of the animals and the responsible use of genetic tools. And as our ability to manipulate DNA grows, so does our responsibility to use that power thoughtfully and with clear oversight. With that, we've completed this chapter. I really hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.